We can best appreciate the cognitive purposefulness of single cells by looking at the most complex assemblage of cells that we know, namely, of course, the human being. 20 million years ago, humans began to emerge from the world of the apes, and 8 million years or so ago, the very first hominids appeared. Sahelanthropus chadensis is known from 7 million years ago. And as time went by, humans began to emerge. We had the first pre-humans 2 million years ago, recognizable as humans, and of course modern man dates back 250,000 years. But what of the future? Now everybody is convinced that we're going to be superseded by, of course, a robot. It's not a new idea. Eric the Robot dates from 1928, almost 100 years ago. The Duke of York, who later became King George VI, was due to open a Society of Model Engineers at London's Royal Horticultural Hall in 1928, but withdrew from officiating. So this prompted the creation of the very first robot. Eric, he's recognised as the first modern robot, was constructed that year by Captain William Richards, a First World War veteran and secretary of the Society of Model Engineers, with his friend aircraft engineer Alan H. Reffel, both distinguished in World War I. The robot was able to stand on command and address the meeting with some pre-recorded words. That's the first robot, and it does date back almost a century. Eric was followed by Alpha, the modern robot, in 1932. He was constructed by the British inventor and engineering professor Harry May. He could respond to verbal commands, and he was introduced in the autumn of 1932 in Brighton, England, where it captivated the audience and rather startled May by firing a gun that he held in his hand and shooting May through the fingers. Alpha could respond to verbal commands.
Meet Alpha the robot, constructed entirely of metal, but controlled only by the voice.
Fire! How tall are you?
6 feet.
6 feet.
Then we came to Electro, who was built for the New York World's Fair of 1939 by Westinghouse at Mansfield, Ohio, built by engineers J.M. Barnett and Jack Weeks Sr. 7 feet, 2.1 metres tall, and weighed 265 pounds. Electro was quite a sight.
I'll be very glad to tell my story.
Throughout the last century, the emphasis was always on the body as machine. It became a pervasive motif. Indeed, this diagram, showing how the body could be paralleled by a factory, was even copied by China and appeared in one of their major teaching textbooks. The notion pervades us all that inside the human is a complex machine. I mean, have a look, for example, at Vitruvian Man, this extraordinary drawing done by Leonardo da Vinci about 1488. Leonardo would little have known that his superb drawing was going to be copied in the modern era, this time showing man's conversion to a machine. The idea of the human machine continued with Thomas Hobbes in 1651 Leviathan. He wrote, "For what is the heart but a spring, and the nerves so many strings, and the joints but so many wheels, giving motion to the whole body." Then, of course, you had L'Homme Machine by the materialist philosopher Julien Offray in 1747. The Human Machine by Arnold Bennett, published in 1908, who kept all his machinery wonderfully clean and well organised, but neglected himself physically all the time. You had Rochebras writing on the human machine in France. Megan O'Geilin, a transhumanist, a word that was coined in 1814 by Henry Carey, and is now used to mean how humans become extended by machinery. George Bridgman is the artist who taught Norman Rothwell, and his book in 1939 conceived of the body as a machine. 2018, Muller and Schaefer, How Machines Challenge Humans. And then Dorleen Kindersley, with a DVD to accompany it, The Human Amazing Machine. Fritz Kahn in 1927, The Human as an Industrial Palace. It was actually made into a movie by Henning Lederer in 2010. Quite a remarkable piece of animation. Inside Lederer's machine you can see the various portions of the brain working very hard. There is a mechanical heart vividly portrayed in this wonderful animation. The stomach, the intestines, the whole excretory system. Everything designed to show that the human body is really nothing but machinery. Now, some years ago I was invited to write a book for children that came out in a whole range of languages on the human body. And there I introduced the idea that no, humans are not machines. A living system is nothing like a machine. Machines are essentially digital and need to be repaired. Humans repair themselves. Whatever the human body is, it is not a machine. And yet wherever you look, the notion persists that cells are like little transistors.
This is a cell. So are all these. They work together doing important stuff, like keeping your vital organs running and whatnot. The hitch? Like you, your cells get hungry. Feed them with Centrum micronutrients, designed to nourish and revitalize you at the cellular level, restoring your awesome on the daily. Centrum, feed your cells.
But no, these are not cells. And yet, even at school, the notion of cells is taught in an extraordinarily oversimplified fashion.
Every living organism is made up of cells. A human sperm cell has to be able to swim, while the cells that produce the peacock's tail must produce beautiful coloured pigments. Sometimes cells can exist on their own, such as a simple bacterium or a single-celled organism called an amoeba. But mostly, cells work together to form more complex multicellular organisms.
But of course, these are not cells. Now, the scholars amongst you will of course at once have said, no, the peacock's feathers are produced by the diffraction of light, by features on an essentially colourless feather. The peacock's feather does not produce any pigments at all. But this notion of cells as being simple and things you can't see is absolutely everywhere. That was for school children. This is for adults.
If scientists succeed in uncovering all the secrets of the cell, it can be a tremendous benefit for all humanity. Cell research is crucial to understanding how life works. Cells are like tiny machines that need energy to function. The possibilities are enormous as scientists continue learning more and more about these building blocks of life. For the cell's life is part of our life.
The Wallenberg Foundation has funded this splendid research institute, but the people who work there can't even take video down their microscopes and show you what a real cell is like. Those are nothing like cells. They're what I call jelly beans, and they pervade the media. Look online for living cells, let's do a quick search, and all you see are those jelly bean models. There is this belief that you can't see cells, cells are mysterious, and virtually everything you see shows almost anything other than a normal living cell. It's not true that cells are entirely invisible. You can see a human ovum with the naked eye, and should you come across a Tradescantia plant, you will see there quite clearly little staminal hairs in the middle, sprouting from the centre of the flower. And can you see each staminal hair looks like beads on a necklace? Those are single cells, and you can see them with the unaided eye. Under the microscope you begin to glimpse something of what I'm talking about. The detailed intricacy of what goes on within a cell. In the centre, of course, there is the nucleus. And just look at the way in which the cytoplasm is circulating around. There are some cells in which the nucleus itself goes patrolling around, as though keeping an eye on the fact that the cell is working properly. We have no idea why some nuclei do this and others don't. You would think that in this modern era of biosciences, that everywhere you looked you'd see images of microbes, but I'm afraid you don't. You click onto the BBC, for example, where they have sights and sounds of every kind of creature you can imagine. You end up with the bivalves and the clitolata, the earthworms. Of microbes, there is no sight at all. It's almost as though the living cell has been banished from television.
When I first started out, there was so much of the natural world we were unable to film. The cameras, the lighting, the size of the equipment, all meant that the extremely small, the very large, the ultra-fast, or the infinitesimally slow were beyond our reach. In the last 30 years, things have changed out of all recognition. We can now film everything, from the giants of the world's oceans to the smallest invertebrates.
This is the leafhopper of the family Psychedelidae that my good friend David Attenborough was looking at. That may be small in terms of television documentaries, but that insect is composed of some 5 million cells. You can imagine how excited I was one day to hear George McGavin, a great lecturer and biologist, was going to do a programme in which the BBC was showing a revolutionary microscope camera. They said it was capable of magnifying 7,000 times to show the hidden beauty and functionality of Britain's miniature ecosystems. I couldn't wait to see it. And when it came on, this is what we saw.
And then what we can do is we can just wash that out into a cup, and we can collect that to look at under the microscope later.
To actually see them, we're going to have to push our microscope camera to its very limits. What I really want to see, Emilian, is the phytoplankton.
Maybe we can zoom in on this one right here. Yeah, they look like some diatoms. Oh, there he is.
Oh, that is beautiful. Fantastic.
Only glimpses of algae can be seen, and these final sequences are largely 3 overlaid videos of particles of dirt meant to inspire the audience. In fact, they disillusioned everyone. If you grab a bit of seawater and have a look at it, and this is what you'll see. There are some of the marine algae, which are quite typical of what you would observe. And here is Cyclops, little water flea. You can see its single eye on the top left-hand side there. Fascinating. When you think that living cells have created this is an arthropod. It has an exoskeleton made of chitin. And yet just look at that jaw. If you saw a photograph of that, would you think that was from some microscopic little organism a millimetre or 2 in length? I doubt it. You see that the cells manage to create chitinous combs that will filter out even bacteria from the seawater. And look at that claw. Ideal for catching and holding onto your prey. And yet that's all present in a tiny little water flea. On the edge of the claw there is an even finer comb designed to filter out bacteria from the surrounding water. If you need to see what plankton really look like, then let me show you this sequence taken by my late friend Jeremy Pickett-Heaps. This is what you should have seen in that television programme, each of them living their own busy little lives. Occasionally some of them perform what many regard as near miracles. You can sometimes sail across the sea in the tropics and find that the ship's wake leaves a silvery trail glowing in the dark, caused by this little organism, Noctiluca. And when disturbed, it emits bright flashes of beautiful phosphorescent light. Under the microscope you can see quite plainly how it has a flagellum with which it will catch smaller creatures and ingest them. This is a single-celled organism, yet within it is a tiny water flea struggling to escape, but, I'm afraid, to no avail. Here is Coleps, an extraordinary single-celled organism. It's actually endowed with a kind of armour-plated casing. This is the great scavenger of pond water. This little organism can ruffle around to discarded detritus at the bottom of a pond. And if you look at it, you will see how it adjusts its operculum, its mouth, to pick up anything that it wants to scavenge from the surroundings. It has fine cytoplasmic strands sticking out almost like legs. Just look at those just below the main body of that cell. They're moving it around, they're positioning it, they're holding it almost like little legs. It's very hard to imagine that this is a single cell. No, this is no primitive little transistor. This is an organism which is cognitive. It is, if you like, thoughtful. It's purposeful as it makes its way around its substrate in order to find its food. Coleps is a remarkably entertaining little creature, and it's all built from one single cell. This is Lithostomatia, An extraordinarily interesting little protozoan. Again, it's a single cell, but just look at that anterior region, the head, nuzzling around, exploring the environment. There is clearly a sense of purpose and decisiveness, and this is obviously a cognitive cell. It lives its own busy little life. Most people never see it. Most people wouldn't even know that it exists. And yet all the time, there it is. Paramecium is a single cell. You can see it has a sculptured surface as we change the focus. Inside it is its gullet. This exquisite video micrograph taken by Rob Burden of Canada reveals paramecium in all its detail. And let's look very closely. All organisms that live in water tend to take in more water than they need. They must therefore have some kidney apparatus to get rid of the surplus. And this is a close-up of the kidney apparatus. It's speeded up about 25 times. You see, radiating ducts carry all the water to a central balloon-like bladder, which then comes to the surface and pops and returns the excess water to the pond in which it began. You may think to yourself that a mountain stream is clear because it's free of microbes, but no, it's because it's full of microbes. If you touch the stone of a clear mountain stream, you'll find it feels slimy. It's covered with vorticellular organisms like these. They open out, each one a little bell-shaped cell, which sets up a vortex in the water and absorbs all the passing bacteria. These wonderful micrographs by Rob Burden make us realize just how intricate, just how involved, just how purposive are living cells. They detect their environment, and clearly they know what they're doing. Sometimes these vorticellular organisms form a teletroch and go swimming away through the water to find a mate, or sometimes to find a new place in which to settle down and start a new colony. This is Stentor, photographed by Jeremy, a wonderful trumpet-shaped organism. Do you see how it sets up that little vortex? And the textbooks will tell you that Stentor just sweeps things in and consumes them. But it's more selective than that. Stentor detects what it wants to take in and absorbs them. Things that it doesn't want, it pointedly ignores. Quite how such selectivity is carried about is beyond us. Stentor will also break free and swim. Here's one of Rob's pictures of a free swimming Stentor as it makes its way through the pond. It'll be going to find a place where there aren't too many Stentor, where there will be fresh food to gather. And Blepharisma, beautiful shot in phase by Rob. A lovely shot of this very complicated little creature, a single-celled organism. You can see very clearly how it's taking in food through that gullet. And notice too that large round bubble-like bladder, the contractile vacuole, which stores excess water above and beyond what the cell needs for its own metabolic purposes. Metopus is a fascinating little creature in its own right. Again, you can see the gullet through which it feeds, its beautifully sculptured cell wall. Indeed, the notion of organisms like Metopus encouraged Will Wright to create microbes in a computer game. He's with Electronic Arts and the man who invented SimCity, that very famous computer game of a decade or two ago. This was when Will launched it to the public for the very first time.
I'm going to show you a toy today that I've been working on that really I think of more than anything else as kind of a philosophy toy. In playing this toy, does it bring up philosophical questions in you? This game's called Spore. I've been working on it for several years. It's getting pretty close to finish now. And you actually start this game kind of in a drop of water. So here we are at a very kind of microscopic scale, swimming around. And I actually realized that cells don't have eyes, but it helps to make it cute.
I know cells don't have eyes, he says, but it makes it kind of cute. Cells don't have eyes? Oh, yes, they do. Let's look at Uroglena. Here's a little clutch of Uroglena, beautifully photographed by the late Hilde Cantor Lund. And here's one on its own. You see its little red eye spot near the top? Of course this microbe has an eye, and if we take a section under the electron microscope, you can see very clearly the structure within it. But look at this picture of Phacus by Jeremy. An incredibly beautiful little creature. You can see its eye spot under the high-power microscope. And just look at how complicated that structure really is. Some organisms behave in an extraordinarily interesting way that we cannot begin to explain. Eutreptiella, this curious little protozoan, it circulates a blob of cytoplasm up to the front and then back to the posterior for reasons that nobody can begin to grasp. Jeremy took very good pictures of Euglena. Here is Euglena, nuzzling its way through the detritus at the bottom of the pond. See the eye spot there on the upper right, it's quite clearly visible. If we take one of these euglenoids and look at it really closely under the microscope, you can see the beautifully sculptured cell wall that surrounds it. And on the right, its red eye spot. If you showed that picture to a kid who'd never seen a microbe, he would say, well, yeah, that's a little creature of some sort, it's some sort of alien. You can see its mouth, its nose, its head, its eye spot. But this is a single cell with purposefulness, ladies and gentlemen. And do not be deluded by notions that single cells are simple. Take one of these euglenoids and trap it under a cover slip, as Rob has done here. And there you can see it just sitting there, waving its flagellum. This is the flagellum that will carry it on its journey through the pond. And just look inside at that hidden intricacy that we have yet to unravel. And these are creatures, of course, that live in the sea or live in fresh water. Some live in blood. This is leishmania, causes leishmaniasis, transmitted by sandflies. It can take years before this organism, this parasite, suddenly appears and makes people ill. Well, there are two, alongside a huge white cell, a leukocyte, in the middle of the field. And notice that the one on the right, followed by the one on the left, are broken down and destroyed by that white cell and absorbed into its structure. This is how white cells in the blood protect us all from disease. They're protecting you at this moment. You very likely inhaled some little organism, some germ, some bacterium, a staphylococcus or something, which a white cell has identified and absorbed. This is a beautiful sequence taken by Jeremy under time-lapse of a white cell. And if you look at this example, there's a dozen parasites around it, but notice one after the other, they adhere to the white cell. They become captured by the white cell. The white cell breaks them down and absorbs them into its own body. If ever there was an example of purposefulness in living cells, it's in the white cells of human blood. Rob took this very nice sequence of amoeba. Now, amoeba are extraordinary things. If you put a culture of amoeba in a petri dish with some actively swimming little ciliates, come back the next morning and have a look, and the ciliates have all gone. They've been consumed by the amoeba, though quite how they do it has always remained a mystery. Rob Burden actually managed to capture the moment at which an amoeba had just seized a ciliate. Here you can see the organism on the top right, and it seems unable to escape from the grasp of that amoeba. The amoeba closes its pseudopodia around so that the little grazing ciliate is now trapped in a bubble of its own, and it will be digested and absorbed into the system of the amoeba. Amoebae are purposive, and video micrographs like this show them doing what they do best. Many microbes make a skeleton for themselves. I mean, here, for example, under the scanning microscope is a picture of a circular diatom. That shell is made of silica, glass if you like. They secrete silica to create this wonderfully supportive endoskeleton within the cell. This is Actinotychus, photographed by David Williams at the Natural History Museum in London. Trillions of them grew many millions of years ago. And if you go to Lompoc, California, you will see white beds of what looks almost like chalk. But no, it's mounds of circular diatoms. Put a bit of that white rock under the microscope, and there you can see the broken shells of circular diatoms. Now, of course, because of their structure, they make a really good mild abrasive. Indeed, diatomite is often used in toothpaste. Toothpaste diatomite was used in many toothpastes and has been for over a century. Let's look at some living algae. These are desmids. Take a little picture at Microsterias. This is an exquisite little creature, but you can actually see how it will divide in half. Jeremy Pickett took this time-lapse video of one of these cells. It's hard to think how a thing like that could split in two, but Jeremy revealed exactly how it does it. You see, each cell, once the nuclei have divided, each cell sets out to secrete an exact copy of the missing half. And in that way, one microstereas becomes two. They really are extraordinarily beautiful. We've seen those circular desmids. Let's look at those elongated Nosterium. It takes 12 hours to undergo sexual reproduction. Do you see how the cells are putting out little bulges towards each other? We don't know how they detect their presence. We don't know how they manufacture the bulbs. We don't know how those bulbs eventually join together to form a little chamber in which sexual reproduction can take place. But once that little chamber has formed, the cells migrate inside and will secrete a protective spore wall until spring comes and they can hatch out. Diatoms are single-celled creatures, but some of them do live together in colonies that can slide against each other, giving the impression of an entire organism moving from one place to the other. Clearly, they sense from one to another what each one is doing. This is coordinated action, but it really is a remarkable phenomenon to view under the microscope. Now, I dare say in school you also came across Spirogyra, and here is Spirogyra undergoing sexual reproduction, the male filament at the top, the female filament below. You see how the two cells create a tube running between the filaments, and the male cell then propels itself down through to fuse with the female. It's all very well saying this is a mechanical process, but how do they detect the male and female filaments? How do they detect each other? How do they know how to create that cellulose cell wall? How do they manage to do it? How do they dissolve the original cell wall and then create a beautifully smooth and perfect canal through which the male cell can merge with the female? Once they've merged, they will form resistant spores that last through the winter. And note that some of the cells are fated never to lose their virginity. If they don't match up with a cell on the opposite filament, they're just stuck there. Oedogonium is a very interesting little organism, and here a male Oedogonium cell has managed to locate an oocyte, the female cell. It has joined with the cellulose capsule that covers the female cell. It forms a hole, it forms a canal, and then, with a clear sense of purpose, it migrates through the hole into the oocyte where the nuclei can combine and a new sexual zygote can result. This is incredible complexity, and nobody has the slightest idea of what goes on. Here we have Chlamydomonas, which also is taught at school. A circular single-celled chlorophyte alga with two flagella. But they also have a sexual phase in the life cycle. During the autumn, two cells will swim around each other, and if they approve of what they see, they will swim until their anterior portions conjoin, and you in essence end up with a figure-of-8 shaped cell with 4 flagella. And in time, the two cells fuse, the nuclei unite, and you end up with a spore that will survive the winter and emerge again next spring. But how they detect each other, how they know what to do, is entirely beyond us. Bob Burden took this very nice sequence of Blepharisma, a ciliated protozoan. Here are two that have fused together, and it is extremely hard not to see a romantic embrace in these two cells. Yes, I know you always think to yourselves, well, that's just anthropomorphism, isn't it? But it's anthropocentrism that concerns me much more. The fact that we think that everything must be based on what humans do. After all, if humans experience passion and lust, why should not the single cells of which we are comprised? They are us, after all. What they do, we do. Whenever there are graphic simulations online of cells, they always turn out to be plastic and somehow mechanical. This is one of the best. It really does look like a sort of a living cell until you get inside, and then you discover that what you've got are scanning electron microscope pictures of bits of dead cells lurking in the background. The real cell, of course, is voluptuous. This is paraholistica. Moving through a pond with a clear sense of direction, a sense of purpose, and a desire to seek out what it wants to find. Yet you can never find these on any television program, nor are children ever shown them in school. Indeed, now that social media pervades all of our lives, we can see that this falsified jelly bean idea of the living cell, this simplified notion that a cell is just a little mechanical device, is all pervasive.
One day, this lecture hall will be made of code, and though they're virtual students, what they'll learn together is real. The impact will be real.
But no, that's wrong. What this man is holding is nothing like two living cells. Students are being taught a simplified mythical view of life, a view that discourages them from ever wishing to look into the majesty of life in any greater detail. In fact, the study of the living cell is the one area of science that still has yet to be fully addressed.
Aaron Bernstein, doctor and research associate at the Center for Health and the Global Environment at Harvard Medical School, has been studying the ties between the natural world and human health.
Really, we're utterly ignorant about the microbial world. In fact, it is the last great unexplored frontier in life on Earth.
I was always fascinated by such things when I was a kid in school. This is a spectrometer, not a microscope. But throughout my life, I was devoted to developing ways of studying cells and filming them down the microscope. I actually obtained a bomb-aiming camera that had been used on Spitfires during World War II. They were selling them off, and managed to take early images of some of these cells. These are Vorticella. These are actually free swimming cells of Vorticella, moving independently through the water. At Chicago, with Jeremy Pickett-Heaps, we gave a live demonstration of the organisms you'd find in any little pond that came from water in a pond on the roof. Because there's so much mystery about cells which there needn't be. Even a hand lens will show you those little pinpoints of light are single cells. Put them under the microscope and you begin to get some insight into what life is really like. We can take a cell and see within it chloroplasts. They were once independent green algae which have been absorbed by green plant cells to provide them with a source of solar energy. So much for plant cells. What of animal cells? These I made as a student, and this is an example of a rat fetus, an extraordinarily beautiful sight under the microscope. Everybody should be shown sections like this. You'll never understand humanity until you understand the cells of which humanity is comprised. Under higher power, you can see the cerebellum at the back of the brain looking like a little wrinkled walnut. And if we go to higher power still, you can make out these single cells that comprise this embryonic part of the brain. There is a single cell isolated so that you can just see how large the cells are. Here's a second one, and a third. We can separate single cells and you can get some impression of the size of cells relative to this early brain. It's this sense of proportion that everybody needs to have. That section, showing the kidney, the umbilicus, the whole of the tail, and a midsection of the brain, has attracted some attention, and indeed was an award winner in a recent photomicrographers competition. People will say, don't you just look at cells under an electron microscope? Yes, of course you do. This is what they look like. This is a cell under an electron microscope. Several things. It's been sectioned, it's dead, and it's only black and white. This is a section taken at Radboud University in the Netherlands. But if we take a living cell and use selective dyes to bring out the contrast within some of its internal components, this is a picture by Matthew Daniels at the Wellcome Institute, and you get some sense of the complexity that's going on inside there. You want to look at living cells, and this is one of Jeremy's sequences of a living cell in phase, and do you see how the extremities of that cell are what we call ruffling? They're sensing the environment. They're probably searching for another cell. These are cells in culture, and normally, of course, cells grow in layers of tissue, and this cell may well be trying to feel out for its neighbour. Under high power, you can see this ruffling effect at the extremity of the cell. All living animal cells do this. There's the nucleus of the living animal cell, and you can see the little mitochondria dotted about it. Notice that they are moving. They move along in a quite purposeful fashion. Mitochondria evolved from free-living bacteria, and now they act as powerhouses within the cell, providing the cell with a source of energy, while the cell provides the former bacteria with protection and a secure environment. Cells have these fine channels running through the cytoplasm. There is a mitochondrion, and you see how it's quite deliberately moving by itself. These mitochondria, having once been bacteria, they divide as bacteria would divide. In a way, they're independent from the cell. Yet they provide the cell with all of its source of energy. Here you can see the way in which these little particles inside the cell move along predetermined channels. It's like watching traffic on a distant highway. But they're not doing this for nothing. There is a purposefulness behind what they do. These are the extremities of amoebae that you would find growing in woodland. And just look how busy is the world of tiny particles, these minute organelles inside each living cell. The whole notion of cells as simplistic, like little transistors, is absurd when we look at the nature of life under the high-power microscope and see it in all its intricate, majestic complexity. Here is the nucleus, and just look at its attendant mitochondria and little fat storage globules of food all around it. Such intricacy, such wondrous, choreographed complexity. When cells divide, it's always taught very simply. That's how a cell is shown. You see the chromosomes, they're pulled apart by little spindle fibers, and bingo, you end up with two cells. It's all made to look mechanical, simplistic, and easy to model. But in real life, it's nothing like that at all. Mitosis of the living cell is a sequence of complexity and refinement. Here is the nucleus of a cell, photographed by Jeremy. And you will see how the chromosomes, which are invisible in the normal cell, in the interphase cell between divisions, you just don't see the chromosomes at all. But they condense and form when the cell is going to divide through the process of prophase. Then the two cells pull the chromosomes apart so that you now have two sets of chromosomes. A cell wall appears between the two, and now you've got two nuclei and two cells where once you had one. Here is very early prophase of a cell nucleus, and just watch how the chromosomes under time-lapse can be seen to condense and actually form. You can see how the spindle begins to generate, whilst the chromosomes became shorter and thicker and eventually are pulled apart so that two nuclei are going to form where there was one before. This is speeded up about 20 times. But just look at the planning that goes on, the purposefulness within each cell. You can see that the cell is going to divide, but there's a chromosome over on the left which is left out of the pattern and is suddenly pulled in at the very last minute by the spindle. It's as though the cell as a whole recognized it was lost and knew it had to draw it into place. There are two other chromosomes over on the left in this other cell, and the chromosomes are clustered together for metaphase, ready to divide, but those chromosomes are lost. One is pulled in, but the other one seems lost and lonely and remains on its own. Those two cells will not divide in future. The formation of the cell wall between two cells is remarkably complicated. Take an end view of this dividing cell, and this is how the intervening cell wall that will separate the cells begins to form. And do you see how the connection between the cells gets smaller and smaller due to all that activity in the cytoplasm around? Just look what's going on. How can we begin to model this in simplistic terms? This is incredible complexity, beautifully photographed by Jeremy. That is how one cell becomes two. And there are the two cells, photographed by the Nikon microscope company. There are the two cells, freshly divided. Here's an amoeba that I just captured the other day. This is not under a special microscope. This is under a student microscope, and it's not speeded up, it's at normal speed. And there you can see this little blob of jelly. They say in school how an amoeba is just a shapeless blob of jelly. Well, a shapeless blob of jelly would dissolve away into the water. A shapeless blob of jelly wouldn't know where it was going. A shapeless blob of jelly doesn't have a sense of direction, as this amoeba clearly does. Under phase contrast, here is an amoeba. And just look at how it is exploring its surroundings, investigating its environment. And those particles inside, they are performing a function by going where they're going at a predetermined time. That is a free-living amoeba. And of course, such amoebae also inhabit us. We saw earlier a white cell dissolving protozoan parasites in the blood. Well, here is a famous piece of video taken by David Rogers at Vanderbilt University in 1959. And it shows a single white cell under time-lapse, speeded up about 20 times, and it's chasing those bacteria. In some way it senses the bacteria. In some way it knows that its destiny is to attack those bacteria, to engulf them, to inactivate them and to digest them. That is an amoeba, ladies and gentlemen. It is not controlled by the brain. It is not altered by hormones. There is nothing in the body controlling it. It is independent, but it is an essential part of you. And how about euplotes? One of my favourite little protozoans. Single-celled organism dashing about here as though it is intent on a mission. We can explain so easily how a human arm works. I can demonstrate a human limb with a couple of sticks of firewood and an elastic band. But there's no way in the world in which any of us can model how these transparent extensions of the cell's cytoplasm act like little limbs. This is complexity which we have no capacity to understand. There is a little organism that has managed to grab hold of a ciliate and is in the process of digesting it. But the ciliate has a mind of its own, and with a sudden thrust it manages to escape from the cell and return from whence it came. Here is euplotes, and you can see how it feeds, and notice how it packs away those algal cells that it's going to digest whilst rejecting others. We've no means of knowing why it's making that selection, and we certainly can't understand how it does it. Have you ever seen a bird bath or a shallow pool look as though it's rusty in the autumn? People think, well, that's because it's made of reinforced concrete and the iron is leaching through and going rusty. No, it isn't. The red colour is because of this little organism, Hematococcus pluvialis. Isn't that absolutely exquisite? These are little cells surrounded by a ball of jelly, a little bit like the eggs of frog spawn, and propelled by two flagella. There's not rust, it's microbes. And now let's have a look at some of the most remarkable protozoa of all, amoebae that make a home for themselves.
Not the kind we learned about at school, was the shame. I only wish we did. This is a shell-building amoeba called Difflugia. It is often found, if any of you are amateur microscopists or have young readies who are, you can often find it by squeezing the water out of a clump of wet moss. Difflugia often lives amongst moss. And this little amoeba builds a shell for itself. Here are its pseudopodia. This is the little creature itself, and here is the shell that it makes. It goes around and picks up sand grains and glues them together to make this beautiful little flask. Now that's an idealised drawing that I've done there, so let me show you a real one. This is one photographed under the microscope. This is the mouth end, as it were, and there's a little spike on the end. And just look at the way that it has fitted together all of these grains of sand. That's as much skill as you would get in somebody building a stone wall. The shape of it.
Those little shell homes that the amoeba make are quite remarkable. I was very surprised to see these nice little table lamps on display in a recent market. They're made in much the same way as the amoeba makes its shells. Here's one example. Let's have a look at it closely, and you can see how beautifully it's constructed, except of course that it's got one part missing. I've never noticed a mistake like that in an amoeba. But let's have a look at how that man-made lampshade compares with what an amoeba can produce. And there is the shell of an amoeba. This is produced by a person, a craftsman, someone whose dexterity we admire. This is made by a single-celled amoeba, picking up little bits and sticking them together. Here's another one. Interesting, isn't it? Quite remarkable how these simple little blobs of jelly can do it. After all, if we look at a drawing of a caddisfly larva on the left, makes a home for itself, it's quite similar to the little shell that Difflugia the amoeba makes, except that a caddisfly has got eyes and a brain and muscles and claws and cement glands. That Difflugia is just a shapeless blob of jelly, isn't it? These are how I showed them in a recent lecture, and a little while later, I don't know whether somebody from the production company was in the audience, but they turned up on the BBC.
When an amoeba splits up, who gets the house?
Oh, I see.
Who gets the house?
Well, yes,
I thought the whole idea was that they split, they get two houses. It's some sort of perfect world of...
Do you know what sort of houses amoebas can build? I mean, they're single-celled, and it's just part of the amazing nature of all living creatures is that even incredibly simple ones, like an amoeba, you don't get much simpler, can actually build a protective house. They assemble grains of sand, and they make that protects them. Even to the spikes. Those spikes are deliberate. They can... Isn't it incredible?
Is that loads of grains of sand as they pull around?
Yeah, each one of those. It's about the size of a full stop on an average newspaper.
Wow.
It's tiny.
That's insane. We've never heard that before.
That's a goal.
That is actually quite interesting.
Hooray! What happens is the amoeba goes along as a single-celled organism, and the odd bit of sand it absorbs and saves up, and some it uses to build this house. And then when it's time to split, it takes away the house, and the half it leaves behind has got all the building materials left, so it can quickly build its own house. So that's how they split it when they have a split.
Why have we never heard of that?
I don't know. I suppose really.
They never mentioned that.
There's been a conspiracy to keep this information from us.
Let's listen again to what those comics said. I find their words very revealing.
Why have we never heard of that?
I don't know. I suppose really.
They never mentioned that.
There's been a conspiracy to keep this information from us.
Here is the physicist Brian Cox on television with Jonathan Ross talking about Europa, Jupiter's moon.
It's got a liquid ocean underneath it, which is probably about 100 kilometres deep.
So if you fish in there and...
well, there could be microbes. Many people think there'll be at least microbes around. And the great thing...
Microbes don't count. We want things with arms, eyes... I know. That's what we're interested in.
But microbes would be a start.
No, they would... Microbes!
No microbes. It's almost like a mantra. You may wonder why I've never done a series about microbes on TV. Well, I have asked. I once put a proposal to Mark Thompson when he was Director General, who put my proposal over to a producer, George Entwistle. Well, they did make the programme, though they didn't involve me. And this is how the programme began. And what did they use to exemplify living cells? Those bloody jelly beans again.
The story of the cell is the most powerful story in science. Cells do the work for us. It's the theory of everything.
A later programme came with Mysteries of the Unseen World, and I thought, well, we're bound to see some single-celled microbes here, but once again, they were nowhere to be seen.
It let us peer into a world we'd never seen before. Suddenly, we could see creatures in common pond water that we didn't even know existed.
These artemia are each composed of about 100,000 cells each. And suddenly a holographic microscope was announced online. That did look as though it was exciting. It was going to shine new light on microplankton. And what did they show? Computer graphic models. And why is it that they never show living microbial cells? If you want an example of clear purposefulness in a living cell, then just watch this ciliate as it goes browsing through the pond water. It encounters a diatom, which it considers is unsuitable for lunch, but a little further on it suddenly sees a ciliate that it engulfs. It wolfs it down, it dives upon it, like a cat catching a sparrow on the lawn. Although nobody in Britain has yet taken the bait for showing single cells, I've often discussed them in other countries. Here we were in Singapore.
And lecturer Dr. Brian J. Ford from having a long-lasting impact on science.
Well, he has done research on mad cow disease and even had laws changed because of his discoveries. Now, we had a chance to speak to him when he was in Singapore to deliver a distinguished visitors lecture at the Singapore Science Centre.
Well, one of the key studies that you have done, and that's made really headlines in the world of science, is intelligent cell. What did you mean by that?
Cells do extraordinary things. If you look at cells under the microscope, which of course people don't do these days people are looking at the minutiae, they're looking at the molecular genetics of cells, they're looking at cell biology, they're looking in the tiniest way at the smallest bits of cells. When we were in school, we all learned of amoebae as being the simplest, most lowly form of life. But there are some amoebae, and I'm doing studies of these at the moment in England, there are some amoebae that construct a home for themselves. They pick up things like tiny specks of sand and cement them into a beautiful vase-shaped house in which they live. And sometimes you can look at cells under the microscope and see that they've been damaged. And the way that they repair themselves by actually carrying out reconstruction work is much more than just a simple chemical system. Some cells within the cell have a tiny eye.
You know, when science is explained so easy in terms that we can easily understand, isn't it fascinating?
Which all brings us to the ultimate question.
Can a single cell think? The biologist, Professor Brian Ford, is giving a lecture at Churchill College, Cambridge today. He is on the line to, I hope, explain. Professor Ford, good morning.
Morning to you, Jim.
We're talking here about
that thought and data processing in the brain goes on at the synapses, the junctions between brain cells. I don't believe that. I believe that thought is going on within the nerve cells. The nerve cell is the most highly evolved cell that we know, and it cannot just be switching on or off like a transistor. I don't see us, you see, as bodies divided up into cells. I see us as great communities of independent, almost free-thinking cells that come together to produce the effect that we know of as human life. Well, Jim, you've gone suddenly almost speechless, which I have to say is uncharacteristic for you.
Well, no, I mean, just, after a description of what is the nature of life, it's a question of where you go next, really. Professor Brian Ford, we know you're lecturing at Cambridge today. That really was extraordinarily interesting conversation. Thanks very much indeed.
There are many definitions of intelligence, but they all involve solving a problem that has never before been encountered. And I want you to look at this single cell of the rhodovite alga antithamnion. This was captured on film by Jeremy in Australia. He has taken some filaments, some cells of antithamnion, and has cut one in half with a little steel needle. And you can see it's completely broken and destroyed. But the adjacent cells in some way sense what has gone on. Look at all that cytoplasmic activity. This is speeded up 20 times. And the cells reoccupy the damaged cell wall. Not only that, but they then minimize the dislocation between the cells and produce new cell wall material to seal it. Watch it again. Here is the damaged cell wall, damaged, empty, broken cell wall. It is reoccupied by the adjacent cells who have sensed the damage that has happened. They manage to repair the cell, to reinstate it to life, and to mend the damaged cell wall. Filaments of antithamnion could be broken in nature. There might be a rock fall, a rush of water, a dinosaur stepped upon one and snapped it. But however that happened, the two broken ends would end up a long way away. The only place in the world where they're going to end up adjacent to each other is on the microscopist's slide. Antithamnion has never experienced this before, yet this lowly alga managed to work out what to do. That is intelligence in a lowly single cell. So next time you glance at a little stagnant water, don't think to yourself, oh dear, keep away from that, you'll probably get some nasty disease. Don't let the children go near it. No. What you find there is actually a universe of life. So we have seen cells with a clear sense of destiny, a sense of purpose. And I want to leave one important thought with you. My view of life is this. Everything we do is rich in resonances of what the single cells of which we are comprised will do. Whatever we do, our cells can do. You might think, well, I build a home, so can an amoeba. Those microscopic cells can see, can sense, can solve problems, can manifest intelligence. We are comprised of single cells, and there is nothing we can do which single cells in their own simple way aren't doing already. We amplify the potential of single cells. Because we are made of trillions of cells, we can do things which on their own they cannot do. But in principle, every one of their actions is mirrored in ours. We are our cells. What our cells do, we do. My sincere thanks to so many good friends and colleagues, and also to you for inviting me to come and speak to you today. I hope after hearing my words you will never look at single cells in quite the same way ever again.